go ahead and get started. We have an hour for this. So without further ado, here's Dr. Beaudet.
Hello, my name is Art Beaudet. I've been studying Angelman syndrome for a couple of decades now. I basically do human genetics as trained originally as a pediatrician, but now most of my work is in genetics research, and I've been chairing a large genetics department for a long time now. So I think you should all be able to see my screen, and I have a pointer too I think you'll be able to see. And you see a title of Ube3a ATS targeted antisense oligonucleotides as therapies for Angelman syndrome. ATS stands for antisense, as we'll be discussing. I just put this here. This is an abstract of our Angelman Syndrome Foundation funded research grant, which is available on their website and is the work that I'll be talking about. So there are some favorable aspects about Angelman syndrome compared to some other disorders or some other circumstances. One has some disorders where the brain does not grow properly, and even by the time of birth there are very severe changes that would seem to be irreversible if the brain doesn't develop properly. Angelman is a more favorable situation where the brain appears relatively normal early in life, and normal neurologic exam is often present at birth. I mean, Angelman children often are not noticed to have any neurological problems in the first few weeks of life, although in retrospect many of you will have experienced things like feeding difficulties and so on, that things weren't perfectly normal, but if you take a child to a pediatrician, they will examine the child and say that the neurologic exam is relatively normal. And then if you do imaging studies of the brain, certainly early in life these are usually normal. And if you do microscopic examination of the brain, and this can be done either in Angelman mice or in patients who die from some cause, the brain is relatively normally formed at a microscopic level as well. So it looks like the brain is pretty well formed and is intact, and maybe there would be the possibility of changing something that might allow it to function relatively normally. So now you see a slide with a bunch of red and blue configurations that are intended to be chromosomes. The red or pink chromosome indicates coming from the mother, and the blue coming from the father. And over here you have a normal situation, and this arrow here that I'm pointing to indicates that the Angelman gene on the mother's copy of the chromosome is turned on. And at least in the case of neurons or brain cells, the father's copy is turned off. And I've got this little arrow here pointing upwards, which is the antisense. So it turns out that this portion of the DNA is actively being copied, but in the opposite direction to what would be normal to make the Angelman protein. Now, in the other panels here, we have the sort of cartoon depiction of the various forms of Angelman syndrome here, indicating a deletion, so the mother's copy of the gene is missing. Here, a mutation, so the mother's copy of the gene is there, but it has a 1 or 2 letter change in the gene that inactivates it. Here we have so-called UPD or paternal UPD, so 2 copies of chromosome 15 from the father and none from the mother. And here we have an imprinting defect where the mother has contributed a chromosome, but it's behaving like it thinks it's from a father, and it has the Angelman gene turned off. Now what you'll see is that in all of these cases there's at least 1, and in one case 2 copies of a normal paternal chromosome. And you may be familiar already from other work in the field that Dr. Philpott's group developed the term unsilence and asking, suppose we could just turn on the father's copy of the gene. In any of these situations, this would be potentially helpful. Now, I've got a now further depiction on the bottom of this slide that shows here UBE3A. This is the intention to think about the gene being copied in the normal direction to make the Angelman protein and prevent the presence of Angelman syndrome. And over here, coming from the other direction, we have the antisense. And what we have is this collision model, what we call, is that the antisense bumps into the sense, and then they basically, nothing goes anywhere, and you don't get any Angelman protein made. So we'll be talking about this, and I'll show you various versions of this line of thinking. So here I've put this little engine that is sort of spitting out Angelman protein, these little red blocks. And you could imagine that this engine is like this polymerase here. The polymerase is the thing that's actually copying RNA. And you could imagine that each of these polymerase signals up here, red ones going in this direction and blue ones going in this direction, could be like this little engine that's cranking out RNA for the region that it's covering. I don't know if when you all see my screen, are people seeing the notes from the webinar information, or should I close that, or is it fine the way it is?
Each one has their own on their own screen, so you can just go ahead and leave it. Okay,
so it's not interfering.
No, they don't see yours.
Okay, sorry. So now I'm showing, again, a similar situation where we have the maternal chromosome at the bottom here will start, it makes the UBE3A, which is the Angelman protein, and I've shown the little engines chugging along here producing the little red blocks. On the top, we've got the paternal chromosome, and it starts to make the Angelman protein. It starts chugging down the track, but on the paternal chromosome we have this antisense coming in the other direction, and it works such that the collision takes place about halfway through the Angelman gene, and so the little engines going in this direction are not able to make any of the complete Angelman protein. So you have this collision going on the father's chromosome and no output of the Angelman protein. Now, there are some other things that we might be a good time to note, that up here are a number of Prader-Willi-related genes. And so what we'd like to do actually is to turn on the father's copy of the gene, but also leave on these Prader-Willi genes that are in blue up here, because if we turn those off, we'll now start to create other problems along the lines of Prader-Willi syndrome. And so what I'll show you is the strategy that we're pursuing shuts off the antisense here, but leaves all these other things intact. So the oligonucleotide approach I'm going to talk about allows us to be very precise and to come in and change things at a very specific site, which leaves most of the other genes doing their normal thing. Now I've here introduced these X's here that says that in Angelman syndrome this is not functioning, so you now have deficiency of these little red blocks. So a student in my laboratory, Linyan Meng, has been working on the antisense, and she started out with the hypothesis that this functions to basically turn off or suppress the father's copy of the gene. And so she wanted to find out some basic information about this, and she looked at what is the antisense transcript, and what she found was that it's an RNA that is made that stays in the nucleus. Most RNAs go into the cytoplasm to make mRNA or messenger RNA and protein. And what's the function? I'll show you some of her data indicating that one function of the antisense is to turn off the father's copy of the gene. And how does it do that? I've already kind of given you a hint that it's this collision process that turns it off. And then can we use the antisense in some way to develop therapies for Angelman syndrome. So after she learned some about the some about the RNA, she then undertook a genetic experiment to try to insert a stop signal here on the father's copy of the gene. And this stop was intended to stop the antisense coming in this direction and allow the full copy of the Angelman protein to be made from the father's chromosome. So I'm going to show you what happens when she did this. And actually, I'm not going to be able, for reasons I'll explain in a bit, to show you much of the oligonucleotide data. But in fact, it will behave a lot like having this stop signal here. It's a way to maybe, another way to put another kind of a stop signal at this point. And the findings with the oligonucleotide data are in many ways similar to the findings I'll show you with this stop signal being put in here. So here's just a little display that in a normal or wild-type mouse, the Ube3a antisense is made at a certain level and it's just fixed arbitrarily to 1 here. So this is the normal amount of antisense in this experiment. But if she takes the mouse that has the stop signal put in, you can see that the antisense goes way down. And you'll see that in some other figures here in a moment. So she bred mice in a certain way so that the mother would have an Angelman mutation, and the father would have the stop signal on the paternal copy of the gene. And there's 2 copies of the Angelman gene, so you get various combinations of mice. One that we'll call wild type, which is normal, healthy mice. One that will be the Angelman mouse. One that inherited the father with a stop signal, but this is not too much interest to us because that's all it has. It doesn't have the Angelman mutation. And the one that we're interested in most is this that has the Angelman syndrome on the mother's chromosome and the stop signal on the father's chromosome, and we would hope that this mouse would be helped by having the stop signal there. Okay, this is again the cartoon you've seen, and in this case we are going to do it so that the mother's copy of the gene is inactivated as is shown here. The stop signal will be on the father's copy of the gene. Okay, so now here's the evidence of the Angelman gene RNA here that is being shown here, where it says Ube3a. This is Ube3a RNA. This beta tubulin is just a control to show that the samples are well matched. And what you see is, in a wild-type mouse, there's a certain level of the Angelman RNA being made. In the Angelman mouse, it's markedly reduced. Looks almost undetectable in this image. In the stock mouse, it's actually a little bit increased, but this mouse is not much interest to us. It's sort of a byproduct of the experiment. The mouse we're really interested in here is the mouse with the AS stop, that is, has the Angelman change, but has the stop signal on the father's chromosome, and you can see that now the father's chromosome is making some of this Angelman RNA. We know that the mother's chromosome in this mouse can't do that, so this has to be coming from the father's copy of the gene. And this was sort of the brain cortex for the mouse, but if you look at other parts of the mouse brain, it's similar. This is another part of the brain called the hippocampus and another part of the brain called the cerebellum. You see sometimes the Angelman looks a little different, but in general, the Angelman has very little expression of the Angelman RNA, but the Angelman stop mouse always has some of this RNA. So this is evidence that putting the stop signal in there does turn on the father's copy of the gene. And this is just some plots that try to put some quantitation on the bands that you're looking at here. And these are all the wild-type tissues. These are all the Angelman are way down. The stop is up slightly. And the Angelman stop is increased here. But you can see it's not up to the wild-type level. It's up a lot from this level, but not up all the way to the full normal level. So it's about 60 to 80% of wild type when you stop that. Now we don't know how complete the stop signal is. One possibility is that the stop signal is not stopping all of the antisense, and that may be the reason why we don't see the complete level that we see in the wild type. Now this gives you some idea of more visually across the brain how this looks like. Here, brown signal indicates cells that are expressing the Angelman protein, the Ube3a protein. So here we're going to look at this point at the hippocampus, and we see these brown signals coming strongly here from the cells that are expressing the Angelman protein in the wild-type mice. In the Angelman mice, this signal is much less. It's barely detectable. But then in the Angelman stop mice, you can see it's gone back to a very easily detectable level. So you can see wild-type, Angelman plus turning off the antisense. You see that the Angelman gene from the father's copy comes back on. And this is the same in these other, but further down is just different parts of the brain. But again, what's important is wild type is turned on, Angelman is turned way down, and Angelman plus stop is turned back on. Now the father's copy. In this case, here with the wild type, it's the mother's copy that's doing it. Over here, it's the father's copy that's turned on, not the mother's copy. The mother's copy in that particular mouse has been just inactivated. So the question, obviously, if you turn the gene on, does it benefit the mouse in any way? So we try to look at various aspects of what we call phenotypes, which is the mouse's behavior, the mouse's properties, whether the mouse is healthy, has seizures, living, whatever. So what we see here in this slide is that the Angelman mice, shown with the red squares, end up weighing more than the wild-type mice. So there's a slight trend towards obesity. It tends to be more sometimes in the females than the males. And so you can see here the difference between the wild-type mice or the healthy mice and the Angelman mice. Now when we put in the stop mice and the Angelman stop mice, the stop mice, the green signal is of not too much interest to us, but the purple signal, which is these circles here, you see that the Angelman weights come from up high to down lower, closer to the blue, which is the normal. So it looks like the insertion of the stop signal and the turning on of the father's copy of the gene seems to reverse the modest trend towards obesity that you see in the Angelman mice. It's not so common to see obesity problems in Angelman patients, although it can be seen in certain circumstances. But it's certainly not, the obesity is nowhere near as consistent in the patients as it is in the mice. So now we're looking at another test of the mouse's ability to learn. It's a fear conditioning test where the mice are trained that in the presence of a certain environment or noise that they're going to be given an electric shock. And they start out here naive, meaning they haven't yet been trained. And over here they've been trained, and what you see is that the wild-type mice in the blue, they have more freezing behavior. That is, when you signal to them that the shock is coming, they sort of freeze, or they become frightened by the anticipated shock. In the Angelman mice, they don't learn as well, and so they don't seem to be as fearful of the coming shock because they don't remember or understand it as completely. So here the lower peak for the red Angelman mice is an indication of some impairment. And now when you take the mice that have the stop signal and the father's copy turned on, you see there's a improvement and coming back really to the normal range for fear conditioning. So the mice with the stop signal and with the father's copy of the gene turned on show some improvement in this fear conditioning measure. This is an accelerating rotor rod. So there's a rod that they put on, and the longer they stay on, the better. That's an indication of their skill. And there's a learning component, but there's also just a a coordination component to be able to stay on the rod. What you see is that right at the first trial, the blue control mice, normal mice, stay on the rod longer than the Angelman mice, and you see that they get better at it over time if you do multiple trials. The Angelman mice start out lower and they get better over time as well. So now what happens with the other mice? Well, again, the green mice are not of much interest to us. They're kind of a control. But we can see that at the early time points in the early training, the mice with the stop signal and the father's copy turned on do better. They do more like wild-type mice or more like healthy mice. But it doesn't persist, and so there's kind of a partial correction of this. And so it's somewhat encouraging, but you could say, well, we'd like to know more about can we get a better correction, a more complete correction. And I think that that's going to be an ongoing question as we start trying to look at other ways to achieve this using drugs. There's also a marble burying test, which we don't really understand very much, but it's really dramatically abnormal in Angelman mice. You put marbles in their bedding, and normal mice kind of poke around and they kick up the bedding and end up covering up the marbles, but Angelman mice don't do this much at all. So you can see that here's the control mice, and there's this dramatic change in the number of marbles that they cover up. And if you now take the stop mice, they go back to the control level, but again, we're not very interested in the green. You see, again, we see a partial correction. Then now there's some movement in the direction of being more normal, but it's not complete. And so again, whatever it is we're doing, we would ultimately, in these kind of mouse experiments, like to develop conditions where the mice can get all the way back to the normal level and not just partially corrected. So the conclusions from this work were that genetic depletion of the antisense, or blocking the antisense, is sufficient to activate the paternal Ube3a gene and improve some behavioral findings. Phenotypes are just the behaviors. So the question is, could one use a therapeutic method of some kind of drug to achieve this rather than to manipulate the gene in this way? There's not at the present time any easy way to put in a stop signal in all the brain cells in a child with Angelman syndrome, but maybe we can have some other more medication type ways to do this. So the first, I think, really exciting development in this direction came from the laboratory of Ben Philpott, who reported that topoisomerase inhibitors, a particular class of drugs that are used for cancer treatments, could unsilence the dormant copy of the father's copy of the gene. Again, this was in mice, using mice that we had developed and mice that I'll show you a bit more about that we've used as well in the oligonucleotide studies. I think at the time this was, to me, phenomenally exciting, and I guess I actually got kind of excited about how soon we could try this in patients, and I suggested we have some conference calls about this. But in fact, I think the company that's involved with these drugs and some other data that have come along have suggested that topoisomerase inhibitors are a possibility, but they have some disadvantages as well, which I'll mention a bit further. So that was the end of 2011 when that was first reported. Now there's another collaborative study going on that is funded by the Angelman Foundation that involves Dr. Philpott and Dr. Elgersma in Rotterdam. And Dr. Elgersma made a mouse where it starts out with an Angelman type finding, but you can use a medication to switch the Angelman gene on. And so they are collaborating to study this kind of a mouse. And the question that one would hope to address here is, if you turn the Angelman gene on when a mouse is 3 weeks old, will it correct all of the findings, and will you end up with a normal mouse? Or it is possible that there are some development changes in the brain that we can't really see, such that the brain is permanently impaired and you can't convert it back to normal. Dr. Algersma presented some of these data at the Angelman scientific meeting last summer, and I don't want to talk about his work necessarily, but he did present some data that at least preliminarily, if you turn the gene on late, that wasn't necessarily all that beneficial, but if you turned it on earlier, that might be beneficial, and they haven't published that work yet, and I would guess they'll continue to work on this and at some point publish the findings. So some questions that would be coming up in this setting then: could topoisomerase inhibitors be used for treatment? Potentially, but if one had to give them chronically, they are a poison of a type that poisons one enzyme system in the cell. They're used for treatment of cancer cells where the intent is to kill the cancer cells. And they work on many different genes in the body. It turns out they have effects particularly on long genes, and so all the long genes in the cell get impacted by the topoisomerase. And it also doesn't spread around in the brain as well as one might like. So there's room to have some kind of a drug that would be have some advantages over topoisomerase inhibitors. How early would one need to turn the paternal copy of the gene on? We can do studies in mice, but ultimately I think it's only studies in human that will address this. I do think that it would be fairly easy to diagnose all Angelman patients at birth. If it was necessary to start treatment at birth, this would not be a big obstacle. It would certainly be more complicated if one had evidence that one would need to start treatment before birth. One could think about trying to diagnose Angelman syndrome prenatally and start treatment before birth, but I think the best thought for the moment, I think, would be that maybe starting at birth would be adequate, and that we can identify all the patients who would benefit at birth without too much difficulty with current technology. How specific is the treatment? Can you have a treatment that, rather than affecting all long genes or all antisense genes or something, can you make it be specific for the father's copy of the Angelman gene? That's the only thing you want to change, and you want to leave everything else alone to the extent that you can. And then are there other ways to unsilence or activate the father's copy of the gene? So when this student, Lin-Yen Meng, was working in my laboratory, and she's just now moving on to a next step in life, but at one of her committee meetings there was a faculty member who was on her committee who was familiar with work that was being done at ASOs using oligonucleotides. And he suggested that we try this out, and he put us in touch with the people at ASOs, and we began to talk with them and design experiments. And then eventually the student defended her thesis, got her PhD, and has continued for one more year in the lab as a postdoc or postdoctoral fellow. Now, one thing to know about oligonucleotides is that they're little bits of DNA, single-stranded DNA, that are like typically maybe 15 to 25 letters long. And I've put at the bottom of this series of letters that would be some random assortment of the 4 letters, the GATC letters that comprise DNA. And if you make something of this approximate length and you ask for it to find a match in the genome, in general it will have a fairly unique match. And so it won't be 100% specific, but it will be very close. It will mostly seek out and pair up with the region that you have made it a match for. And of course, now we know the sequence of the entire genome, so we can make oligonucleotides that have the sequence near the region that is of interest to us. Now, so you can theoretically make an ASO that will go where you want it. And what we really wanted to do is to go where we had that stop signal in the genetic form of the mice. So we make it with sequences that match to that region where we had tried out the stop signal. And you can make them with modifications such that they will do various things to the RNA or DNA once they reach there. They may damage the RNA, damage the DNA. They may change the way the RNA or the DNA behaves. And I have another slide here that may show a little bit more of that. So this is a cartoon from a publication from some of the people at ISIS. And I guess I intended to provide this reference, but I guess I forgot. I can make access to this published article if I don't have it somewhere here in the remaining slides. Now they have their first project to try to use oligonucleotides to treat genetic disease involving the human brain and spinal cord involves a condition called spinal muscular atrophy, or SMA. In that case, they put certain chemical modifications that they can put on these oligos, and in the case of the SMA, they use this pink version here, which is a particular chemical modification. But you basically make up an oligo of your choice that matches about 15 to 25 bases along the DNA sequence like this. And this is out of 3 billion letters. So you try to find something that will make a match out of 1 in a billion or so kind of possible matches. So this is the way the study was designed. We made a bunch of ASOs, or ISIS made a bunch of ASOs in the region we thought was of interest, which is in here. And so you try out a whole bunch of them. You put one at a time into cultured cells, and you see if any of them do what you want them to do. And in this case, the idea is that the ASO would sit down here and would stop the antisense and allow the father's copy of the gene to come on, given that the mother's copy of the gene would have the Angelman alteration. So we used a mouse, the same mouse that was used to screen for the topoisomerase inhibitors, and this mouse has a Ube3a Angelman protein linked to a fluorescent marker. And let me fill this all in. So what we do is take a second here, be sure I explain this properly to you. So ordinarily, it's set up such that here we have the Angelman copy of the gene with this YFP, which is a fluorescent tag, and it's on the father's copy of the gene, so it's turned off. And it's being turned off actually by the collision process of this antisense coming at it. And so you don't have any of this green fluorescent material to see. And now when you put the ASO in, the ASO can come in. It would come in this region. It would stop the collision coming from the antisense and allow the father's copy of the gene to be turned on and generate this fluorescent protein. Now, at this point, I have to tell you that I had a whole bunch of slides, and we do have data, but there's multiple reasons why I naively thought I would just show you everything, but it turns out, the companies involved, and there are 2 companies involved, both Biogen, IDEC, and ISIS have a collaboration. And these companies want to have certain protection for their intellectual property and patents, and their goal is to get a drug that they can bring to market. In addition, we've submitted a publication to a very competitive journal. In the journal, you have to sign that this work hasn't been published anywhere else, and you know, if this is unique, it's going to be copyrighted with their publication. So in order for us to publish in journals like that, we can't just, I might in certain circumstances be able to show you some slides in a meeting, but to then have them end up on the website where the whole world can come in and look at them is not acceptable if we ultimately want to publish. So I guess there'll be, assuming we do get a publication out, we can then show you all the data that's in the publication and review it with you and explain it to you. I think I'll just summarize a little bit some of what you would see if I was able to show you the slides. So we screened oligonucleotides in a broad region around the sense and antisense region, and we found that some of the ones that were in a position to block the antisense did create the appearance of this fluorescent green protein. And so we could check each oligo at a time, and some worked better than others. We took some of the better ones and looked at what happened in cultured cells if you changed the concentration. And in general, as you had an oligo that worked, the more you put in, the more you turned off the antisense and the more you turned on the sense. So it was doing the kind of things in what we call a dose-dependent manner. We also checked as to whether it was turning off any of the other genes which we didn't want to turn off, some of the Prader-Willi genes, particularly genes called snow RNA genes. And we also checked whether it turned off any long genes that similar to what happens with the topoisomerase. And again, the results were quite encouraging. The oligo, as we might expect because of its sequence specificity, it seemed to have as its major effect to just affect this one region that we had tried to design it to impact. So we also looked at this at the level of RNA and protein, and what we could see in the cultured cells is that when you put on the proper oligonucleotide, the paternal copy, or the copy, did turn on at an RNA level, and it did turn on at a protein level. So it looked like the oligos in cell culture were doing what we wanted them to do. So we then went on to actually inject the oligos into the brain of mice that had Angelman syndrome. And in general, the results were somewhat encouraging. The oligos, and the company ISIS already knew this, the oligos spread out in the brain quite well, much more than I would have anticipated. And they're quite different than topoisomerases in this regard. So if you inject the ASOs into the spinal fluid portions of the brain, into the water ventricle portions of the brain, it gets spread out quite widely across the brain. And we have images of that we hope to publish. In contrast, topoisomerase, when you inject it into the fluid spaces in the brain, it doesn't spread very far. So I think actually ISIS has been trying to develop these ASOs for more than 20 years, and I think that they've done a lot of things over the years to try to make them stable and to try to make them spread around in the brain. And so we benefit, I think, by years of attempts to make better and better ASOs. So we then were able to demonstrate, if we then sacrifice the mice at certain intervals after the ASO administration, and we treated just with one injection into the ventricular space in the brain, we could see that throughout the brain, the paternal copy of the gene was turned on. Again, these were mice that had an Angelman mutation on the mother's copy of the gene, and they had a normal copy of the Angelman gene on the father's chromosome. And we did see that the father's chromosome could be turned on, and that you would see the protein, and that you would see the RNA as well, as you would expect. The trickier part has been, can you correct the behaviors? Can you improve the rotorod performance? Can you improve the learning performances? It seems that correcting the weight gain and or the mild obesity that's present looked encouraging. Some of the behavior tests are partially corrected, and certainly not everything is corrected at this point. So we're trying to go on to additional rounds of experiments to develop a protocol that will allow for the oligos to try to get a more complete correction by trying to find better oligos that want some that work better than others, to try to give them at earlier times and maybe give multiple doses. Right now we've started out with only one dose at what I would say would be a young adult kind of age. So the conclusions that I can't show you the pictures to back up are that the antisense oligos can be used to knock down antisense and to Ube3a in cultured cells and in living mice, that the ASOs targeting the antisense can activate paternal Ube3a in cultured cells in mice, and ASOs injected into the brain ventricles spread through the brain and persist for up to 16 weeks. So the potential advantages of this approach is that I've said the ASOs are quite specific. They don't go all around affecting genes willy-nilly. They tend to focus on the region that we chose to match them up to. There's some evidence that these can be used, and I'll tell you a bit more about human studies in spinal muscular atrophy. And they distribute reasonably well in the brain and stick around for a good amount of time, and they can unsilence the father's copy of the gene. So I've put a few questions in here, and many of you may have many others, but how would ASOs be administered? Bolus, which means by just one shot injection into the spinal fluid in the lumbar space, is what ISIS would intend initially. I would imagine that an ISIS, I'm not speaking for ISIS here, that one might ultimately have administration into the ventricles in the brain, and maybe even have a pump that would chronically administer the drug. How often might they be administered based on what we've seen in the mice and what they know about the spinal muscular atrophy? Maybe once a month to once a quarter. Would they work for all Angelman patients? Well, as I said at the beginning, all Angelman patients have a paternal copy of the gene, so we would think yes if it's really the Angelman gene. Now there are some other things which people say that are second genes, a second Angelman gene or something. So if it doesn't involve the Ube3a gene, it won't work. But for the most part, when people say there's a diagnosis of Angelman syndrome, they're talking that the Ube3a gene is altered. What age would treatment be started? I would say presumably the earlier the better, and that I'm sure that older children will be receiving medication in any kind of trial if a trial occurs, but I would say that if things went really well, probably one would perhaps be thinking about starting treatment close to the time of birth. Would older patients benefit? Possibly, but almost certainly not as much as younger patients. Would ASOs represent a cure? I think it's too early to speculate, but I would say it's not completely off the table. It could prove to be very favorable. How expensive would treatment be, and would it be covered by insurance? The research part is paid for by the companies that are trying to develop the drug and get FDA approval. If it was approved by the FDA, I think it would likely be relatively expensive, but I think for these treatments that are like dramatically beneficial, there's been ways to get them to most all the patients. So if you think about AIDS medications in the United States, most everybody has access to AIDS medications, no matter how expensive they are. And even throughout other parts of the world, there are ways to try to deliver those medications to many, many people. So I would say yes, it would be expensive, but it wouldn't be prohibitively expensive. And I think most patients would have access to treatment by one means or another. So this is, if you want to go on the internet, you can go to a site called clinicaltrials.gov, and I've just searched for ISIS and spinal muscular atrophy, and you can see that they have a series of trials. One is recruiting, one is completed, one is active but not recruiting, active but not recruiting, and enrolling by invitation. The enrolling by invitation is an open-label safety and tolerability study of this SMNRX is their oligo for spinal muscular atrophy in patients who previously participated in some of the other studies. This is the publication that I referred to earlier, so I do have the title and the authors and this PMID number. If you go into PubMed, you can pull this up, and I would guess that maybe the foundation could put this article on its website. So this is a story of the early work with the spinal muscular atrophy, and hopefully it will be a great precedent. So in spinal muscular atrophy, this is a cartoon from that publication showing how it works. These are the ASOs, which would, instead of having 4 pegs, would have maybe 20 or so. They would enter the cells. They would find their way into the nucleus. And in the case of the spinal muscular atrophy, they don't inactivate anything. They, in fact, alter the splicing or the piecing together. And you can see over here that this RNA gets assembled with 6 linked to 8 and 7 is dropped out, and that causes a problem. So the ASO in that case changes the splicing so that now you get 7 included and a more normal 6, 7, 8 kind of RNA. So it's a little bit different in spinal muscular atrophy as to exactly what the oligos are doing, but I think there's enormous benefit to Angelman research that the spinal muscular atrophy work has gone on. So this is Linyan Meng, who I've mentioned a number of times, and she's really done every bit of the work that's been done at our end. There are people at ISIS who are doing other parts of the work. And this is an acknowledgement slide with Linyan's name and some of the other people here, and the various funding sources. Okay, so I will stop now, and we're at 50 minutes after the hour, so somebody now can join in and tell us what's next.
Well, thank you very much, Dr. Beaudet, for your presentation this evening. We have had an abundance of questions that have come in during the course of your presentation, and so we're going to ask some of these questions of you and look forward to your answers. So the first question, do the antisense stop mice have any phenotypes from overexpression of Ube3a or from loss of the antisense?
Not that we know of. In fact, it's more the case that I think I mentioned that even with the stop mice, we can't get the expression from the father's copy of the gene up fully as high as it ordinarily would be from the mother's copy of the gene. So I would say we think that we would, if the oligos or the stop signal worked perfectly, it would simply make the paternal copy behave exactly if it was a mother's copy or a maternal copy. So I think it would restore the expression to what would be there if the mother's gene was not damaged. So I think in theory, as far as we can anticipate, it would be a fairly good situation. It's probably going to be more trouble to get it turned all the way on, and there's relatively little likelihood of turning it on excessively. We don't know if the antisense does anything else, but we're only blocking a small portion of the end of the antisense. So we don't think that a lacking antisense is going to be a problem, and we have some data that the other nearby genes that are associated with Prader-Willi, like the SNO RNAs, are not affected by the oligos.
Okay. Another question that came in is, would treatment be effective for individuals with UPD?
Yes, actually, UPD patients are probably in a especially beneficial category because they have 2 copies of the paternal gene. And one might even guess that they would benefit more easily, or let's say earlier. Let's say the treatment started out imperfect and it helped some, but it didn't do the whole job. It would be likely that UPD patients would get more benefit than all the rest.
Okay. This next question is a multi-question, so I'm going to read.
Let me go back a minute, and I don't want to. Okay. I don't want to leave everybody else thinking that's bad. I mean, I think it should work fine for everybody, but the UPD patients, they sort of have a slight advantage. It's not that it's a disadvantage to all of the great majority.
Along that line, I know there's another question in here about imprinting center mutations. Can you also address this in regard to individuals with imprinting center mutations?
Yes, I think as far as we understand, this would work for all patients who have the UBE3A gene involved in any way. Now, if they've got another gene, like if they really have Rett syndrome and somebody says there's similarities to Rett syndrome and Angelman, that wouldn't affect Rett syndrome. But as long as it's what we would consider a true Angelman diagnosis with documentation that the UBE3A gene is involved, yes, they should benefit. There are some handful of patients who seem to have all the clinical features of Angelman syndrome, but we can't find an abnormality of the UBE3A gene right now, and maybe there's some question there. If the UBE3A gene was the one causing the problem, it would probably work. If it was some other gene and we can't figure out, it probably wouldn't help.
Okay. The next question is, in your opinion, why do you think you are not seeing a full or better behavioral rescue by knocking down the antisense in the AS stop mice?
Well, there's a variety of reasons. One might be that there's some permanent damage, and it's where we're coming to the scene too late, and that some permanent damage can't be reversed. I think that that can be addressed in part by trying to treat earlier, and it's very hard to draw analogies from what age one might treat. It is feasible to treat newborn mice on the day they're born, and so we can examine in the mice whether earlier treatment seems to help. Another possibility is we don't think we're completely turning off the antisense and turning on the sense. So there's some combination of maybe there's permanent damage that can't be reversed, maybe we're not coming early enough, and maybe our effectiveness is not sufficient. Obviously, the worst case scenario would be, say, at birth, that there was permanent brain injury that couldn't be reversed. I think there's room to be optimistic at this point, but we just don't know all the answers.
Okay. Would stem cell activated with ASOs be a feasible treatment? Stem cell treatment is continually proving to help in a variety of impairments and diseases.
I mean, I think you could study the ASOs in stem cells, but I think that the prospects of just using the ASOs directly as a medication is far more promising than getting stem cells involved. I think that stem cell benefits are, in most people's opinion, still quite a long ways off. One could imagine, and I think that ISIS has indicated that they have substantial interest in the possibility of trying to do something with Angelman like they're doing with spinal muscular atrophy. They don't have a firm commitment to do a trial yet. We're hoping that they will decide to conduct a trial. But I would think that just using the ASOs as a medication and leaving stem cells out of it would be by far the more short-term approach.
Okay. How transient was the unsilencing of paternal Ube3a following the administration of targeted ASOs?
So we saw the gene stay on as I think something of the order of many weeks, 16 weeks or so. But there'll be some data about that when assuming we do eventually publish this, and I think that also at some point we will look at re-administration and so on. But for right now, the work has been restricted to just a single dose.
Okay. As a somewhat related question to that, is there any complication with the ASOs crossing the blood-brain barrier to obtain sufficient concentration of the ASOs in target regions to sufficiently turn on the paternal copy?
So we're not asking the ASOs to cross the blood-brain barrier. We're injecting them sort of on the inside of the blood-brain barrier. Now, they could cross and come out, but they don't do that very much. So we're going to put them on the brain side of the blood-brain barrier, and we don't want them to leak out too much. And ISIS has been optimizing oligos to try to do this kind of thing, to do lots of different things. But I did say that the ASOs do spread around the brain quite well compared to certain other kinds of chemicals. So in general, they seem to spread out reasonably well. Whether one would, how often one would dose and how high the doses would be, I would say the ability to reach all parts of the brain looks generally optimistic compared to many other drugs, which are much harder to get delivered. And topoisomerase, by comparison, I'm told, doesn't spread into the brain very well if you put it in the ventricles, and the ASOs do a much better job at that. So hopefully I've addressed the question.
Okay. Is it correct that only a few ASOs have made their way to market, and what obstacles are most rate limiting to this? And the third question is, what approach can you take to overcome these obstacles?
So this ISIS company has been around, I believe, since the late 80s. They do have one or two, and maybe more. I don't really know. But they don't have a lot of drugs on the market that are based on antisense. But they've been investing enormous amounts for many, many years. They've made a lot of progress. I think some of the compounds that are available may be to treat viral infections and such. There's no approved antisense for treatment of a brain condition like this. The spinal muscular atrophy is the furthest along. I do think, though, that they are quite encouraged. If you look on the internet and Biogen IDEC and you search Biogen IDEC and ISIS, you'll see that they announced a few months ago, or maybe 6 months ago now, a collaboration where the two companies would be investing a lot of money into trying to develop oligonucleotides as treatments for brain disorders. And I think the companies are a lot more optimistic today than they were, 10 years ago. So I would say a lot of the obstacles have been overcome, but we don't have much data yet, either in animals or humans, as to whether there's still some unappreciated obstacles that haven't been overcome. But you could hope, I think, that in the spinal muscular atrophy that there will be evidence of benefit in the not too distant future, and that drug may really work out to benefit those patients significantly or substantially. So I think the companies feel like they've overcome many of the obstacles, but of course it's very expensive and very time consuming to bring any new drug to market. And a relatively unusual class of drug like the oligonucleotides also has got its challenges. So that's my view of it. And at some point, maybe people from ISIS could try to address some of these questions.
Okay. I just lost my internet connection here. Sheila, are you on the line? Can you read a couple of these questions, because I've lost my internet connection here?
Sure. I'll just do a couple more, and then we'll start to wrap it up. I don't know, Dr. Beaudet, if we sent you some of the questions that we weren't able to get to tonight, if you would be willing to go ahead and answer those, and then we can send them out to all the attendees later. One of the other questions is, has any AS kid ever been treated with, and I'm sorry, I'm not good at pronouncing it.
Topoisomerase.
Yeah, if so, what were the results?
None has been treated to my knowledge. I know there's been some discussion about the possibility of treating one patient, but I don't think anything has gone on, and I don't know if anything will go on.
Okay. And then we'll just do one more for right now. Was there any observation regarding absence of seizures in the work you did with the mice?
No, I didn't talk about seizures. The mice do develop seizures, but it's very tricky. It's depending on the certain background strain. But one of the things we would hope is that we could eventually develop a model whereby the seizures that are developing in the mice can be prevented by the ASOs. That's what we would hope. But the seizure studies in the mice are somewhat tricky, but it would be, I'd love to be able to say that, we've used the ASOs to prevent the seizures in the mice, the Angelman mice. That's something we'll be trying to do.
All right, good. Well, I think that's all we have for now. Dr. Beaudet, we definitely appreciate your time and doing this at night. Again, the session has been recorded. We will put that up on our website later, and then we'll also, Dr. Beaudet, will you answer the rest of the questions and maybe send them out to us when you have some time?
Within limits, yeah. Okay.
Of course.
I'll try.
All right. Thank you very much. Have a good night, everybody.
Thank you very much.